So we spent some time discussing aerobic cellular respiration, which requires oxygen. Now we're going to spend some time talking about what if oxygen isn't available, how do we still make energy, and we call this process fermentation or anaerobic cellular respiration. So most cellular respiration does require oxygen to produce ATP. In fact, that's the most efficient way to get a lot of ATP is in the presence of oxygen. However, glycolysis doesn't need oxygen to occur. Glycolysis happens in the cytosol of a cell, and it can happen with or without oxygen. So in aerobic conditions with oxygen or anaerobic without. If there is no oxygen available, glycolysis will occur, but instead of those pyruvate molecules crossing over into the mitochondrial membrane, into the mitochondria, they will instead stay in the cytosol and go through the process of fermentation or anaerobic respiration. Through this, we can produce some ATP, but not as much as if we were under aerobic conditions. Anaerobic respiration does use an electron transport chain just like aerobic respiration, but the final electron acceptor, instead of being oxygen with hydrogen ions to form water, could be something different like sulfate. Fermentation uses substrate-level phosphorylation instead of an electron transport chain to generate ATP. So the first step of anaerobic respiration is glycolysis. Glycolysis forms your two pyruvate molecules. The big idea behind glycolysis is regenerating your NAD+, your electron acceptor, so that glycolysis can again donate electrons to it. So no matter what process you have after that, you want to regenerate your electron acceptors. There are two types of fermentation that we will talk about, and that's alcohol fermentation and lactic acid fermentation. In alcohol fermentation, you start with a glucose molecule, and the glucose is broken down by glycolysis in the cytosol to two pyruvate molecules. As the glucose is broken down, it loses electrons. Those electrons are picked up by NAD+ to form 2-NADH. This guy has those electrons. In order for glycolysis to continue occurring, you are going to need to have somewhere that you can drop off those electrons, and that's where fermentation comes in. Also, as a result of glycolysis, we form a net ATP of 2 ATPs. So the 2-pyruvate are converted to 2-acetylaldehyde, and that's where you lose CO2. So in alcohol fermentation, we do get carbon dioxide as a product. This is used in beer making. It's what gives us our carbonation. The 2-acetylaldehyde is then converted to 2-ethanol, which is an alcohol, and in the process, we use 2 electrons in order to create that final product of alcohol, so that frees up your electron carrier so that it can pick up more electrons to process more glucose through glycolysis. So this keeps glycolysis and keeps the cycle going. It doesn't make much in the way of energy, but it keeps the cycle going and the organism doesn't die. The other type of fermentation is lactic acid fermentation. So now we have glycolysis, which breaks down a glucose into 2-pyruvate, and that pyruvate is reduced to NADH. So NADH picks up those electrons from that conversion from glucose to pyruvate, and you also form lactate as an end product. There is no release of CO2. Some fungus and bacteria use lactic acid fermentation to produce cheese and yogurts, and human muscle cells also produce lactic acid to generate ATP when they run out of oxygen. So if you're exercising and your muscles don't have enough oxygen, they are going to switch over to lactic acid fermentation just to provide enough energy to get through that period where oxygen is scarce. So here's what it looks like. Glucose is broken down to 2 pyruvates through glycolysis. We form a net of 2 ATP. 2 ATP are invested and 4 paid off for a net of 2 ATP gain. The 2 pyruvates are converted into 2-lactate. That's where your lactic acid fermentation comes from. In the process, the electrons that were released and picked up by NAD+ to form NADH are used to power the reaction converting 2-pyruvate into 2-lactate. So what this does is, in that conversion from 2-pyruvate to 2-lactate, is it frees up your electron carrier again, so it's free to pick up more electrons in glycolysis, so as glucose is being broken down. So it continues the cycle. It does not provide very much energy in the form of ATP, but it continues the cycle long enough to keep the organism alive while oxygen is scarce. So it's a short-term remedy for organisms that are not obligate anaerobes. So let's compare and contrast cell respiration and fermentation. Both processes use glycolysis as their first step. This happens in the cytosol. You are taking glucose and breaking it down into 2 pyruvates. Your final electron acceptors in fermentation would be an organic molecule such as a pyruvate or an acetaldehyde. And in aerobic respiration, your final electron acceptor is oxygen, which pairs with hydrogen ions to form water. In cell respiration, we produce 38 ATP per single glucose molecule. In fermentation, because we don't go into the mitochondria and we don't use oxidative phosphorylation, our net production of ATP is only 2, which are produced in the glycolysis stage. So there's a huge trade-off of how much ATP you're making depending upon whether oxygen is present or it's not. I mentioned the term earlier without explaining it. Obligate anaerobes are organisms that carry out fermentation or anaerobic respiration and cannot survive in the presence of oxygen. So if they're in oxygen, they die. They have to go through the process of fermentation after glycolysis. Yeast and some other bacteria are what we call facultative anaerobes, and these guys can survive using either fermentation without oxygen present, or if oxygen is present, they can switch over and go through cellular respiration to get their energy. So they tend to have the best of both worlds. Here's your big overarching picture. We have one glucose molecule. The glucose, no matter whether oxygen is present or not, will be broken into 2 pyruvates as a result of glycolysis. Those 2 pyruvates, if no oxygen is present, are going to go through either lactic acid fermentation or alcohol fermentation, depending upon what the organism, which mechanism the organism used. If there's oxygen present, the pyruvates cross over into the mitochondrial membrane and they get converted to acetyl-CoA. Acetyl-CoA then enters the citric acid cycle, and eventually you have oxidative phosphorylation, which occurs later on. So no matter what type of organism you are, it's very likely that you go through glycolysis. Based on this, we know that glycolysis probably evolved in ancient prokaryotes before there was oxygen available in the atmosphere. And oxygen did not become available in the atmosphere until about 2.7 billion years ago. We know we have fossils of bacteria that have been around for 3.5 billion years. So we know that they had to have some mechanism to give them energy before oxygen was even present. So glycolysis was selected for. Another piece of evidence we know, just dating glycolysis as a process, is knowing that glycolysis does not require membrane-bound organelles. So you would see glycolysis more in those early prokaryotes, those organisms that don't have membrane-bound organelles, and glycolysis and fermentation do not require membrane-bound organelles to occur as they happen in the cytosol. We know that glycolysis and the citric acid cycle are not only used in cellular respiration to make ATP to do work in the cells, but we know there are also major intersections in various catabolic pathways, so breaking down of organic molecules, as well as anabolic pathways or biosynthetic pathways. Catabolic pathways can funnel electrons from many kinds of organic molecules into cell respiration. So what that basically means is we can get energy from things other than glucose. Catabolic pathways are those that break down molecules. So we can break down not only carbohydrates, but we can break down lipids, we can break down proteins. Glycolysis will accept a multitude of different carbohydrates. The one thing about proteins, though, is that they do have to be broken down into amino acids, and your amino groups by themselves can feed glycolysis or the citric acid cycle. Either way, though, because of the breakdown of those macromolecules, we can get electrons to funnel into oxidative phosphorylation and make a ton of ATP and release a bunch of energy. Fats are a little tricky. They are digested to glycerol, which can be used directly in glycolysis, and then also they're fatty acids. So that glycerol comes off, the fatty acids come off, and those are used to generate acetyl-CoA, which is a necessary reactant for the citric acid cycle to start. Fatty acids are broken down using beta oxidation, and that's what gives us our acetyl-CoA. If you were to compare gram per gram of a glucose compared to a gram of fat, an oxidized gram of fat produces more than twice as much ATP as 1 oxidized gram of carbohydrate. So fats, you've always been told, store more energy than carbohydrates or any other type of macromolecule. I love this diagram because it shows us where exactly the macromolecule and in what form enters into cellular respiration. So proteins have to be broken into amino acids, and then they enter your cell respiration at the pyruvate stage, the acetyl-CoA, and the citric acid. So you're getting electrons from all of those stages that will eventually power oxidative phosphorylation, the electron transport chain, and chemiosmosis. Carbohydrates you know really well because we talked in terms of glucose as we talked through all of these examples, but your glucose molecule enters glycolysis, is broken into pyruvate, then acetyl-CoA, and so on. Fats, your glycerol is going to enter during glycolysis, as we just mentioned, and your fatty acids are used to make acetyl-CoA, and that's where they enter cellular respiration. As we briefly mentioned, your body does need to make larger substances, larger molecules, by using smaller molecules to do so, and we call these anabolic pathways or biosynthetic pathways. And so the small molecules can come directly from food, they can come from glycolysis, they can come from the citric acid cycle, and then we can piece those together to form larger molecules.